Now the room is open for discussion, and yeah, we're accepting some questions. But hey, probably we can get started with our questions. So I have a question to Mauri, and can you repeat the importance of having biomarkers in your research, non-biomarkers in your research?
Yes. Researchers try to find biomarkers in autism and ADHD, but it's very complicated because those are heterogeneous group. It means that there are other comorbidities like eating disorder, depression, anxiety, sleep disorder, and many other comorbidities. So it's very difficult to find those biomarkers. So there are no biomarkers yet, and assessments are done clinically. So usually interview or questions to the parents or the caregiver of the patients, and they're very subjective. So there are a lot of mistakes, and often ADHD is misdiagnosed with depression or anxiety. So in our department arrive people that are high functioning, that they have a normal IQ, and for maybe 10 or 15 years try to get antidepressive or treatment for anxiety that never work. So we need the neurocognitive data to identify the better diagnosis.
Okay, sounds good. So we still have 12 minutes left, so we're going to accept questions. So please introduce your name, affiliation, and also probably your research topic, if possible. So please. Oh, that way? Sorry, please.
Hi, thank you very much for your presentation. So my question goes to Maurizio, and I think you sort of started speaking about what I wanted to ask about. So in addition, sorry, my name is Phyllis. I am from the Liggins Institute in New Zealand, and I am a PhD student studying perinatology. So in addition to having multiple comorbidities, we also know that these autism ADHD, they're actually a spectrum. So I'm trying to wonder how helpful the assessments are going to be when we're trying to sort of personalize them because of how differently they present for something like autism. So by doing an assessment and realizing that this person is autistic, it doesn't say much because it's such a wide spectrum. So how do we move on from there with your assessments?
Yeah, the idea is that not all the patients need to go through that, but only the most complex cases. The high functioning, the one that have a high intelligence, the one that clinically you cannot really differentiate. So we used to jump in only when it's a complex case. It's not for all the patients, otherwise it's going to take too much time. But when, for example, you have some traits and maybe we're not really sure about it, then it's required this kind of support.
Okay.
Okay, next, please.
Hi, my name is Charlotte Bishop. I'm from Macquarie University in Sydney, Australia, and I'm studying a Bachelor of Clinical Science. So I have a special connection and interest in neurodivergence. And so, Maurizio, my question goes to you. So did you find that there was a difference between male and female children when you did your study?
No, I didn't look at that, but we have a project ongoing to see how is the gender difference on neurocognitive data, yes. But on adults, on children, we didn't check that.
Because to further expand on that, I've seen that there is a difference, especially in, as you said, you are going to continue to look at the difference between adults, male and female adults as well.
Mm.
How would you apply your scale, as Phyllis said before, because it's such a spectrum and it's more likely that women are going to go ahead undiagnosed later on in life. So how would you apply that same model to detect that sort of thing in women, in adults?
Yeah, we know that we have more women than men in ADHD because of hormonal differences, and as I said, we didn't really identify and study that yet, but it's one of our projects. So we have a master student from the Netherlands that has to develop that. So I cannot really reply, but is one of our next study that started to start next week, actually.
All right, thank you.
Okay, please, next.
Hello, my name is Fa. I'm from Thailand, and now I'm doing dental medicine research in University of Geneva, Switzerland. I have a question for Dr. Take for the clinical implications of your technology. Thank you for your presentation, by the way. So the first question is that, like you mentioned at the beginning of your presentation, that actually X-ray can be focused and like the blur image can actually be like clear. Why isn't that already being utilized in the clinic? Is it because like when you beam the X-ray, it's like the concentration of the radiation dose is too high? Is it right?
Okay, so actually, no focusing is performed at the clinical environment, just because, yeah, it's a radiation damage first. So, and also, the X-ray source at the hospital is too weak to implement this type of a system. So, if we have strong X-ray sources, like as shown at the beginning of my presentation, like large research facility, then it's possible right now. So still, my system is under development, and I hope that I can implement my system to the clinical or lab-based X-ray source in the future.
Oh, so yeah, that's my second question also, because now it's only doing like you only X-ray on like cellular level, but it's not in actually in clinical. And you probably see your technology in clinical implication in the
Yes.
future.
Yep,
Right.
definitely. And so, of course, large facility is quite limited in terms of access. So I want to spread my system, if possible, to laboratory, and hopefully, yeah, I want to implement. I can implement my system at the hospitals maybe in 5 or 10 years. I'm not sure.
Thank you so much.
Thank you so much. So please.
Hi, I'm Alina. I'm at ETH Zurich, and my background is in analytical chemistry and mass spectrometry. And I have a question for Debsankar. I was wondering if you, or maybe I missed it. Did you also look at structurally similar compounds to serotonin, like dopa or epinephrine? And did you see similar results? And then also I wanted to ask, because I read that serotonin is most abundant in the gut, if you think maybe the research that you did is also applicable to gut cells?
Yeah. So I only showed the results for serotonin, but we found that dopamine and norepinephrine, so these three are monoamine neurotransmitters. So all of them actually behave similarly. The effect is highest for serotonin and the effect like lowest for norepinephrine. But we also tested other neurotransmitters, for example, GABA, glutamate. Those doesn't show this effect. So it's a special effect of the monoamine neurotransmitters. And the second question, yeah, so the receptor that I talked about, Y4 receptor, so that's in the guts. And yeah, and also it is related to food intake, and people have already previously found that serotonin is somehow related to food intake. So yeah, there is a connection between.
Cool, thanks a lot.
Okay, please.
Oh, I have another question for Debsankar. I'm Claire. I'm a Newton International Fellow at Cambridge, and I had a question about the total internal reflection spectra or images that you showed, and I was just wondering how you get the images. Were there fluorophores in there? I mean, you were showing that it would come down and attach, and you would get a signal from that.
Yeah.
Could you just elaborate on that a bit more?
Yeah. So so in total internal reflection fluorescence, what happens is that you only get the fluorescence when it is very close to the cover slip. So it's like 100 nanometer penetration depth. So when and now the vesicles are labeled with a fluorophore. So rhodamine labeled vesicles, and then the vesicle, when the vesicle comes and like comes close to the surface, and the surface, on the surface there is your bilayer. So when the vesicle comes, you can see that vesicle has come.
I see, because there's a fluorophore
Yeah.
on the vesicle.
Yeah.
Okay, cool. Thanks.
Okay, we can accept one more question.
Hi, my name is Jaewoom No. I'm from South Korea in post-tech, and I have a question for Mr. Takenori.
Okay.
From your speech, I've come across that you're designing lenses in X-rays to like avoid problems such as aberrations and other stuffs. And I was wondering if you have any intentions to incorporate deep neural networks to like enhance your images.
Okay, so that's a good question. And so applying neural network to the X-ray image is actually an ongoing project because basically, that's a good question. Hold on.
So like your network can help you design the lenses and furthermore enhance the performance of the images and so on.
Okay. So actually, I'm not too sure about the answer because I'm using X-ray focusing mirror instead of lenses. But I guess it's possible, and there are some research about aberration compensated lens, which whose shape are calculated by optical mathematics and also neural networks. So some optimization is ongoing, and I think this is the kind of hot topic in X-ray physics field. Did I answer your question?
Yes, thank you.
Okay. So we are almost running out of time. So thank you so much, guys, for paying attention to us.